All right, this is our second program on adaptive immunity. We're going to go over the humoral immune response. Okay, last time we went over cell-mediated immunity. Now we'll do the second part, humoral immunity. This is the production of soluble proteins that fight infection called antibodies. Remember, adaptive immunity has three big features. First, it's specific. It targets the molecules of the invading bacteria, virus, or whatever organism. We call those molecules antigens, and they generate an immune response. And that response is only targeted on those antigens. Secondly, this response improves over time. We have better defense against repeated infections, and this defense comes more quickly, and it gets stronger, much stronger, 100, 1,000 times stronger. There's more cells involved, more proteins involved, and so generally we only get sick once because the immune response has memory. And then thirdly, adaptive immunity is tolerant. Okay, only foreign molecules are destroyed, self molecules aren't. And so this system has the two arms shown here, cell-mediated immune response and the humoral or antibody-mediated response. In the cell-mediated response, lymphocytes directly attacked antigen-bearing cells. Okay, in the humoral or antibody response, it's secreted proteins called antibodies. Okay, this is all from lymphocytes. Remember, lymphocytes are just like other white blood cells. They come from red bone marrow. And lymphocytes all look similar, but some of them mature in the thymus and they become T cells. Others don't need to go to the thymus. They'll go into the circulation. They'll mature right in the red bone marrow, and they'll become active in lymph nodes. Okay, but this whole system is the response of lymphocytes. Okay, remember the T cells, they recognize antigens because they develop this T cell receptor. It's made up of protein chains that have constant regions and variable regions. The variable regions undergo genetic rearrangements to produce literally millions of different kinds of shapes that can recognize antigens. And so that's the unique thing. And so this variable region will become the antigen receptor. Okay. So thymus-derived lymphocytes are T cells, all have this T cell receptor that binds to the antigen. They also have the MHC class II receptor, which is kind of unusual. That allows them to recognize other cells that are immune cells. And there's two big types of T cells that we talked about. The helper T cells, they have a marker on their surface called the CD4, and they secrete cytokines to help activate other cells. And then the cytotoxic T cells, they have the CD8 marker on their surface, and when they're active, they directly attack antigen-bearing cells to lyse them and to digest them. Okay, so now we're going to go into humoral immunity. This is antibody-mediated, and antibodies are soluble proteins that are secreted into body fluids. Antibodies are made up of four chains of protein, four protein chains, two heavy chains and two light chains. Okay, the heavy chains have constant regions, but the light chains and the heavy chains also have variable regions. And so these variable regions, a light and a heavy chain together, variable region, that makes up the antigen binding site. And antibodies, you can see, each have two antigen binding sites, in this case, a Y-shaped molecule. And that's the variable region, and so kind of similar to the T cell receptor, but a little bigger molecule, and having this ability to bind to the antibody twice. And both Here they show a green and a red side and a blue and a yellow, but those antigen binding sites are identical. Okay, so what's good about antibody? Well, antibody acts as the B cell receptor, but it's also secreted. It's secreted into blood plasma. It's secreted into our digestive tract as part of our digestive juices. It's in saliva. It's in stomach acids. Um, it's secreted onto our skin in tears, in sweat, and it can fight infections in any of those places, and it can get there pretty fast, and so it can circulate around the body pretty quickly. And when it does find the antigen, it binds to it. And because you have more than one receptor on these antibody molecules, it will cause them to clump together. That's agglutination. It will also cause antigens that are dissolved in fluids to start sticking together and precipitating, so they won't be able to travel anymore. And then for when it binds to surface molecules that are used to like enter cells, it stops those molecules from working, so it neutralizes them. So in the case of viruses, they may not enter cells anymore. Um, when it does bind on a cell that carries antigen, it acts as an opsin, which means it causes phagocytes to become much more effective at engulfing that cell. And it also activates complement, so that it once it's bound to the surface of the cell, it can cause that cell to lyse. And so antibody is really effective. It can kill lots of different things. It inactivates antigen-bearing cells and viruses. It's effective at killing. And you can see here's a space-filling model of an antibody there. Okay, I should also point out there's more than one type of antibody. There's several types. One the type that's made most quickly in an infection is called IgM, shown in the first column there. And so it's very good at fixing complement. It's very good at agglutinating. It actually has 5 of the Y-shaped molecules linked together. So it has 10 binding sites. Very large, works very quickly in blood, fixes complement very, very well. Okay, the second type is shown there is IgG. Okay, this is a Y-shaped molecule that has 2 antigen binding sites. This is the most common antibody in the plasma that's floating around. So most of that immunoglobulins in the plasma are IgG. It's made in a secondary response. It's very good at neutralizing toxins. It's a very good opsin. It also fixes complement, but not as well as IgM. But it's small enough that it can cross the placenta. So mom's blood can protect the baby, or mom's antibodies in mom's blood protects babies. Then there is the third column there is IgA. That has 4 antigen binding sites. It's 2 Y-shaped molecules held together by what's called the secretory component or secretory piece. It's found in secretions, on mucous membranes, in tears, saliva. It's in breast milk, and it can work in the baby's digestive tract. It doesn't fix complement, doesn't cross the placenta, not common in blood, but effective on our mucous membranes. So very, very very, very important that way. And then you have IgE, which has, again, it's got 2 binding sites. This is not really common in blood at all, but it's found on the surface of basophils and mast cells, and it's involved in anti-parasitic activity. It's also in allergies and things like that. And then IgD is just another term for the B cell receptor. Okay, so when we're talking about antibody, then we again are talking about the same kind of production system that we have with the T cells, in that the antigen receptor on the B cell surface has to combine with the antigen that it recognizes, the specific antigen. Once that happens, that B cell reproduces. Okay, it makes exact copies of itself. Those are called clones. And some of these clones will actively start secreting antibody. And they're literally secreting hundreds of thousands of these molecules every second. It's an amazing amount that they secrete. And so they begin fighting the infection right away. Some of the cells, though, reproduce, are kind of set aside for the future. So you produce not only activated B cells that are secreting antibody, those are called plasma cells, but you also secrete cells for future infections. And so the clone expands. You get more and more cells that recognize that specific antigen. And in addition, in later responses, you get class switching, which means that primary response is producing IgM, but later cells can produce IgG and IgA and IgE. And so those types of antibodies generally bind better, and they're better at certain other tasks, like getting to certain places, working on the mucous membranes. And so that's part of that response getting better. So that secondary response in this graph, I've shown it before, and I've kind of talked about it when I talked about T cells, but it's specifically for antibody-related immunity. It's the memory cells in the secondary response that make the response faster and stronger. There's lots of these memory cells around after a primary infection. They're stationed all over the body, and they're capable of doing the class switching. Okay, generally in a secondary response, there's no symptoms, and once we've gone through that primary response, we generally have antibody present in the blood, but there's no real lag time. We're also going to produce much more antibody, maybe 1,000 times or 10,000 times more, and that antibody will bind to the antigen better, and it will be in more places. It will be secreted onto our mucous membranes, in our sweat, in our tears. It'll be in mast cells. It'll be all over the body, and so we get much better. Okay, and so this is the whole purpose of vaccination, is to generate these secondary responses, because generally people don't get sick. I should say a little bit more about B cell activation. It turns out sometimes B cells don't need T cells to help them get going. These happens when an antigen is classified as a T cell independent antigens. Generally, those types of antigens have repeated carbohydrates on their surface. And so they have this repeated carbohydrate feature on their surface. And that is true of many bacteria. And so we don't need T cells for those. But most antigens, it turns out, are T cell dependent. And they require both direct binding of the antigen to the B cell receptor and the signal from the helper T cell, that cytokine. And so there has to be both this communication between the helper cell and that antigen binding. And that's what's going to get the B cell active to go through clonal selection to produce those plasma cells and to produce the memory cells. So we generally need both. Okay, so in summary, adaptive immunity is specific, has memory, and is tolerant. It includes humoral, antibody-mediated immunity, and cell-mediated immunity. The antibodies are specific proteins that can bind antigens in body fluids. We're not talking just blood, we're talking plasma, tissue fluids, digestive fluids, perspiration, fluids on mucous membranes. You'll get antibody in compartments like in other body cavities and things like that. Okay, antibody has a bunch of mechanisms for destroying antigen. That includes binding to it and clumping it together, but it also includes activating phagocytes, activating complement, and so very, very effective at kind of saying, here is something that needs to be destroyed. Okay, activation of antibody-mediated immunity usually requires both antigen binding and cytokines from helper T cells. Okay, so I'm going to stop there. Next time we're going to talk about vaccines, and we'll start talking about things that go wrong with the immune system sometimes. So really important. Okay.